Oh, we don't have Rob yet. Maybe Rob. Oh, there's Rob and there's Jordan. Good. Rob and Jordan, if you have a question, speak up. You can always unmute yourself. I should have said that from the beginning of the semester, but you know. Uh, all right. So I'm gonna cover two topics today. The food contaminants, residues, and chemical constituents of concerns. And then the last chapter, which is very, very short, is extraneous matter. So I will cover them both in this lecture. Okay, so basically we're going to talk about everything that shouldn't be in the food. And they are in the food either due to processing, potentially processing, like generating acrylamide during processing, or something that was added as an adulterant, like melamine, or something that was added like pesticides and antibiotics or allergens. So there are different types of contaminants or residues that shouldn't be in your sample. So, and they can get in our food at different parts of the food chain. All the way when we are producing, whether it's an animal or it is a crop on the land. So we might have the pesticides on the land. You might have the animals with antibiotics. And then during transportation, we might get other contaminations, biohazards, during processing, during retail and transportation. So at any point of the chain, from production to consumers, there are chances of getting these unwanted components in our food. And there are so many categories, unfortunately, and they're unavoidable. You can't have any food product without anything at all in it that shouldn't be in it. So this is old kind of reports, but this is kind of a figure that I like to illustrate that there are hazards that get reported, and these hazards are classified into different categories. So we have the chemical hazards. So chemical hazards like pesticides are chemical hazards. Heavy metal is a chemical hazard. Acrylamide is a chemical hazard. So there, and then you have the toxins, the whole mycotoxins from fungal growth. So, and then you have the microbiology, which is all of those bacteria that can be present in your food. So of course, we're not covering the safety from a food micro perspective, but that's another category of hazard. Another big category that includes fraud, like adding melamine, for example, biological hazards, labeling issues, like sometimes the ink from the label that, do you know why cereal boxes, when you open, there is the inner plastic container bag? That to prevent any ink from the outer plastic getting into your pot, due to the components of the ink that can be hazardous to us. And also, for example, packaging, not only defective packaging, but packaging that has the BPA, the bisphenol A, for example, water bottles. So this wide other category can really have many different things in it. Okay, so the way I categorize this lecture, or at least how I remember I categorize this lecture, is talking about the big ones. So the pesticides, drug residues, antibiotics, the mycotoxins, the allergens, and then we have a whole category of chemical additives and other chemicals. So melamine, adulteration, adding sulfite and nitrite. Where do we add nitrite? Usually in the processing. Where is a common place for it?
Bacon curing.
Yes, curing of meat. Yeah. What were you going to say? Sausage. Yeah. Okay. So it is really added to kind of for preservation, and also it gives a characteristic color as well. Sulfites, people have sensitivity to sulfites. Yeah. So we'll talk about different ones. This is a good table to study from. Table 33, 1 in your book. And I did update my slide. I believe that the slide was updated with the table in it, with this table. If you have a slides from previous that doesn't have this table or similar to it, just look in your book for 33-1. Okay. So what this table tells us, the different contaminants in alphabetical order, and quantitative methods and qualitative methods. So, we talked already about ELISA, PCR, if we want to look at DNA of a particular source, like if you think there is peanut potentially, but not the protein, you do PCR assay to look at DNA material rather than protein material. So most of the quantitation you can see it's HPLC, GC, ELISA. Yeah, so it's mostly GC, HPLC, or immunoassays. In semi-quantitative, we have the letter LFS is lateral flow detection. So you have thin layer chromatography, TLC, and then some electrodes and so on. So it would be good to just have an overview of this table or review this table. But we have general consideration when we talk about residues and contaminants. They're usually present randomly, not necessarily homogeneously distributed. Sometimes they're also present in very, very small concentration. So there are certain considerations we have to do. The sample needs to really be representative of the entire population, especially when we're dealing with aflatoxin. So you might have a mold growth on one part and not necessarily on others, so you don't have that evenly distributed sample or evenly distributed mycotoxins. So in this case, you have to take a lot of replications and make sure you have well homogenized sample. So sampling is very important. If we mess it up, we end up with wrong results. So sampling is very important. Representative sample is very important. And then there is a lot that goes into sample preparation. Oftentimes, there is a series of extraction that we do and purification. So extraction, fractionation, clean up, purification, because you want to concentrate your residue to an amount that you can detect it. So because sometimes the defect action level is very, very low, or the allowed level is very, very low. So you have to really concentrate in order to analyze. So a lot of that sample preparation that leads to concentration. Often you need to derivatize if you're using especially GC to make it volatile and thermally stable, separation, identification, quantification, and reporting. When you choose your method, you want to really know about your food matrix. Do I have high fat content? What solvent should I be using? What are the characteristics of the residues that I'm looking at? For example, is it going to be a polar pesticide or a non-polar pesticide? So I kind of choose the method based on what I know about the analyte, the food matrix, the potential concentration that I have, whether or not I have the capability to do it in terms of feasibility. And then am I doing a multi-residue or a single residue? This is relevant when we talk about pesticides, because crops usually are given a range of different pesticides. So if I want to look at multiple pesticides at one time, I have an approach that's called multi-residue. And sometimes if I want to characterize a particular pesticide, like to register a pesticide, I need to isolate it on its own and characterize it and determine the acceptable level of inclusion or having in the sample. Then the method will be single residue. That means I purify until I get that one particular residue and characterize it. So here's just an expansion of what we talked about. And this basically sample prep, extraction, and then cleanup. We often need to do a lot of cleanup, and we can do cleanup with chromatography, either size exclusion or adsorptive. And or we use something that Gary talked about, solid phase extraction or solid phase micro extraction, depending on the amount of the sample, where you separate based on affinity to a solid phase versus in the liquid phase. So the common one is this in with those many letters, but you're not gonna remember this, but quick, easy, cheap, efficient, rugged, and S for safe. Yeah. So you don't have to remember that. It was just what they usually label those assays. And then you do the accelerated solvent extraction. If you remember, that was mentioned and explained in the FACT analysis chapter, where we said, okay, we use a solvent to increase pressure and temperature so that we can have more efficient extraction before the solvent is evaporated. So we enhance extraction at higher temperature and maintaining that solvent. And then most of the detection and analysis is done by chromatography, mass identification, and immunoassays. So these are the most repeatedly used methods for these kind of compounds. Derivatization is done only when we need to have specific qualities, like in GC, for example. So I'll talk about pesticides. There are very, very many registered pesticides. That means approved for use. I don't know if they're now probably more than 1,300, but this is just to say that there's so many of them. But in order to be approved for use, they should be registered. And to be registered, they need to be classified and understanding at which level they are acceptable to be in food without causing harm. So then they can be registered and be used. When we know that potentially in certain field we might have 4 or 5 different pesticides, we can do multi-residue analysis. We do GC for non-polar, HPLC for polar. Single residue, like I said, when we are really looking at a new pesticide that we need to register, and then there are the semi-quantitative, qualitative, thin layer chromatography, enzyme inhibition, and I'll talk about that in a little bit. But often it's chromatographic method. GC was the method of choice when most pesticides were hydrophobic. And then you have lots of choices of columns, many detectors that Gary talked about. The common one is MS and tandem MS, meaning MS with another MS and more fragmentation so that we can get better identification power. Okay, so HPLC method is for the polar compounds that are not volatiles and not thermally stable, and there's a lot of those new pesticides that are like that. So the columns are dependent on what we know about the chemical characteristics of these peptides. And again, we can use UV detection, fluorescence, but a lot of time it's mass spec. And mass spec, like I said, in order to have better identification power, oftentimes we do MS-MS. That means we take the first ion from the first MS and fragment it even more in the second MS so that we can get more pieces of the puzzle for stronger power of identification. And this is illustrated here with a standard. So with the tandem MS, your spectra is cleaner and better matched with the standard. You don't have that very many noise. So that's what you want to know with tandem MS. You just want to remember that you get more fragmentation, more power of detection when you have a library to compare to. So you have more little pieces of the fragments from the big original molecule to have a very more identification that you trust. Immunoassays, we just explained that, but if we're using immunoassays for pesticides, there are small molecules, so we use comparative ELISA. We cannot use sandwich ELISA. Now, the most common screening method is you use thin layer chromatography to separate. And what they do is usually the pesticides attack a specific enzyme in for pests that are produced by pests, which is cholinesterases. So pesticides inhibit that enzyme, and that's how they kill the pests. So what they would do is you run your TLC, you spray it with the enzyme solution, this enzyme in particular, and then you put it with a substrate. You add a substrate. Wherever you see color, so everywhere you have enzyme, you see color. When you have a pesticide, you don't see the color because the pesticide inhibited this enzyme. So that's how it works. The next one is mycotoxin, and these are categories coming from fungus, fungal growth. So these are, many of them, you're not supposed to have anything in the food. So aflatoxin B, I believe, it's not on the slide, is a genotoxin. Anything that is toxic to the gene should not be, the level should be 0. Other aflatoxins, other mycotoxins, you can have a particular limit. So tolerance level less than 15 parts per billion. So these are categorized, and those tolerance levels are identified for every type of toxin. Method of analysis, it can be thin layer chromatography with fluorescence. It could be HPLC, it could be comparative ELISA, or we can use immunoaffinity chromatography. So you can't go wrong when you say HPLC and MS, for example. You can also use immunoassays. So if you want to give an example, mycotoxin detection, you can say immunoassay comparative ELISA, or you can say HPLC MS, or with UV detection. Drug residues, antibodies. So animals are given antibodies whenever they are sick. They have potential infection. So they are given antibodies. So at a therapeutic level, or sometimes they are given lower levels of antibodies, although they're not sick, but to promote weight gain, sorry, and so they feed them less. So kind of like make them fatter with less feed. But when the animal is sacrificed, slaughtered, whatever you want to give the name, it has to be off antibiotics for a while. And especially also for having milk, it also should not, the animal should not be on any antibiotics. Why is that? Because these antibiotics, this is some families the penicillin family, the amoxicillin, which is also related to penicillin, sulfa, and other antibiotics. All of these can cause, some people have reaction to antibiotics. So cannot consume anything with these antibiotics in them. Other problem, if the milk has any of these antibiotics, if you want to culture the milk, produce yogurt, or then produce cheese, you have antibiotics that it can interfere with the process, the fermentation or the generation of aging of the cheese. The sensitivity and also bacterial resistance. So basically you generate, if the animal uses a lot of the antibiotics, the bacteria becomes resistant and then the antibiotics no longer work. So that's why there are also zero tolerance for antibiotics in milk or in animal meat products. Two common tests that you might want to remember for antibiotics. The one that is screening turbidity. So you would have a media with some bacteria, and then you add your sample, milk sample, or what have you. If it has antibiotics, you don't have growth over time. So the turbidity will not increase over time if you don't have antibiotics. That's one. Another is a common one, which is, we call it the disc diffusion method. So you have your media, you put your particular microbes, and then here there are small discs that you have, let's say, the milk sample in them, a little bit of milk in those. And over time, if you have antibiotics, you don't see growth. You see growth everywhere, but you don't see growth around the disc. So that means you have antibiotics in the sample. The charm test is a very common one where you radio-label an antibiotic that you're looking for, and you have the milk sample. It works like a comparative ELISA. So the radio-labeled antibiotic competes with the antibiotic in your sample to interact on the surface of a bacteria. If you have high radioactivity, that means you have low concentration of antibiotic or no antibiotic in your sample. If you have low radioactivity, that means you have antibiotics in the sample that bound to the bacteria and did not carry a radio label, so they were not detected. So again, methods, GC, HPLC are common, MS is common, or UV absorption is common. Allergens are proteins. So we have 9 allergens now. Sesame was added. So proteins from these different groups can cause immune reactivity in some people. And because of that, we have this Food Allergen Labeling and Consumer Protection Act, which dictates that all manufacturers should say on the label contains peanut, contains soy, contains milk, contains wheat. Or if they don't contain peanut, but M&Ms, they are produced in the same manufacturing line where you have peanuts. So they could say may contain peanut allergy because they were manufactured in the same facility. Allergen. The way we detect those, either immunoassays by ELISA or qualitative Western blot, or sometimes biosensors, where you have sensors with antibodies labeled that can be used. And these protein assays, they have this advantage because when you isolate the protein, depending on the method of isolation before you test, you may not isolate it properly. So this is a protein isolated with some salt in the isolation process. Here you don't have salt, so these proteins are missing. So if you run Western blot on the sample, you won't see reactivity, but that could be wrong because the way you isolated the sample, you missed some protein. Another one is DNA method by PCR analysis, but that could be also a problem if you're using a highly purified protein ingredient in your product, doesn't have genetic material in it, but the protein is there, then these methods are not good. So it depends on what you are measuring, and in what type of product or ingredient. So moving on, I'm going to give you examples of a lot of other residues and contaminants that you don't want them in your sample. So there's a lot. There is a summary table in your book, but I will talk about a few: sulfite, nitrites, melamine, packaging material, furans, acrylamide, benzene, monochloropropane, and perchlorate. So I'll give you examples on how these end up in the product. And there are others that I might not mention that are banned in certain countries, legally limited, and of concern, like the MSG in soy sauce, for example, causes headache for some people. They consume a lot of it. So many of these compounds currently can only be analyzed by expensive methodology that is expensive. So there's continuous effort in developing methods that are highly sensitive and accurate. But all of the methods, or most of them, are chromatography and MS detection, which is really, more expensive, time-consuming methods. The limit of detection is a major issue because you want methods that can have very, very small threshold, because some of these compounds are present at very small concentration. So I'll start with sulfites. So these are used as antimicrobial agents. Sometimes they are naturally present. So they are present in wine, for example, or we can add them sometimes for dried food products to prevent browning and microbial growth. So we are required to put sulfites on the label, especially if we have a higher than 10 ppm. But we have to do that if we have more than 10 ppm, because some consumers have sensitivity as asthma reaction to sulfite. There are so many forms of sulfites that could wind up in food. Not one method can measure all of them, but the most common method that I want you to remember is Monnier-Williams method. So this is the most common method that can be used to measure sulfites, most forms of sulfites. It is the method that FDA refers to for labeling. So it is based on putting an acid, heating it with an acid, converting all possible sulfites to SO2. And then the SO2 is oxidized to produce H2SO4, and then you weigh it. Well, either weigh it gravimetrically or you can measure turbidity. And that's what I want you to remember. So the name Monnier-Williams are the people that obviously found that method. It is FDA referred to for labeling. You add an acid HCl to convert it to SO2. You react it with H2O2 to oxidize it to produce H2SO4. You measure it gravimetrically or measuring turbidity. There are other methods that I'm not gonna go through in detail, but just you will hear about them somewhere else. Ripper, enzymatic method. You can also use chromatography with fluorescence detection. And that HPLC with fluorescence is an approved method by AOAC. I've been talking a lot. I can't, everything is dry, huh? I don't have my water bottle. It's fine. Okay, nitrite and nitrate. So they are used as preservatives, mostly to cure processed meat, gives you that color, and it is specifically, it prevents the growth of Clostridium botulinum. Sodium nitrate can also be present naturally in some vegetables, like celery, for example. There are legal limits for nitrates and nitrites, and the problem with them is they can form nitrosoamines at high temperature. These guys here, nitrosoamines, are carcinogens. They cause a specific type of a blood cancer. Method of analysis, colorimetric method, or a chromatographic method. I'm not gonna have you worry about details here. There are rapid methods. We have ion selective electrodes. I know that Dan talked about some electrodes that are used for mineral detection. So like we have a pH meter, but the electrode will be specific. Instead of the H groups in acids, it will be specific to the ions of interest. There are test strips that can be used as well. So don't worry about the details there. Melamine, have you heard of a few years ago? Maybe you were very, very young, maybe in elementary school, of melamine adulteration. Mostly in the US, it was in pet food. So what happened was, in pet food, you have gluten and you have rice proteins. And from what you know, that Caldol measures amine groups. This is melamine. Look how many amine groups this melamine had. So what they did to give you the protein content that should be in the pet food based on using gluten and rice protein. So they use less of the gluten, less of the rice protein, and they put melamine in there, and the Caldol doesn't differentiate because it's measuring amine group. Even if you do DUMAS, you also look at all the nitrogen. So when those pet food passed through integration, in protein, so they passed detection because you measure protein by Caldol or you measure protein by DUMAS. Nobody knows. A lot of cases of pets happened where pets died from consumption of certain pet food. It does impact the kidney mostly, and it can be fatal. In other countries of the world, it was used in milk products for infant formula, which also babies got sick of it. So what is melamine actually used for? It's an organic chemical used in plastic material. So it's not supposed to be in your food. It was an adulteration. So this is an example where it's not naturally present. It was added to the food to falsify the protein content. I don't think they knew what was going to happen from a health perspective, but those people that adulterated these products, but then it turned out to be a problem. Melamine is detected by HPLC MS or GCMS. So chromatography again and mass spectrometry. There are other methods for detection that we are trying, well, not we, but people are trying to develop that are fast, like IR detection, for example, and immunoassays. This is a very common contaminant in bottled water, but now you might see bottled water that says bisphenol A free. So it comes from polycarbonate plastic, and it causes also, it mimics hormones, and it impacts more the children and the babies, infants. Banned actually from being used in the plastic, or present, depending on the country, present in low limits. Again, LC, GC, MS, immuno, affinity columns, ELISA. This is the the chemical that comes from ink. It's not so recently, I should change that, a few years ago. In cereals, due to the ink migrating to the cereal from the container. Now that's why there's a secondary container in cereal boxes to prevent that situation. Again, LCMS-MS is used. Furans, these are produced during when you have high carbohydrates, you have some acid in the food, you have heat, you generate those furans that are carcinogens. So again, processing these are not added to the food, they don't come externally, they can be produced with processing. They are volatile, so they can be detected by GCMS. Acrylamide, we talked briefly about it. It is a carcinogen and it's generated also for heating. So, and a good example, deep fried potato chips could have acrylamide. Because you have the reducing sugars potentially from the carbohydrate. You have proteins. Asparagine is amino acid in a protein, and then with high temperature, you can generate acrylamide. LC, again, GC, MS. Benzene is a carcinogen. It can generate if you have a beverage that has ascorbic acid and benzoate salt. So you can generate benzene. It's volatile, so GCMS is used with a headspace sampling. Monochloropropane, this can be used when we are creating hydrolyzed soy protein using an acid. So if we use HCl, for example, you're welcome. If we use HCl to break down the protein in soy, we can generate from the residual fat in those proteins, we can generate monochloropropane. So in a protein ingredient, that especially coming from an oil seed, you will always have a little bit of fat in there. And that could, in the presence of acid, form that compound. And it is volatile, so GCMS. The last one, perchlorate, that could be, which is a compound coming from rocket fuel specifically, but it can be in the soil. And if it is in the soil, it can end up in water, in milk. There was cases where it was found in lettuce. So it impacts our thyroid gland and causes hypothyroidism. This is the table. There is an updated table. I didn't put the updated table apparently here, but it's similar table. I'm not sure of the number, but it's in chapter 33 that summarizes all of the compounds, why they might end up in food, why are they of concern, what is the food product that they could be found in, and what's the net. So this is similar to the question that we reviewed in lab earlier. So you want to know what an example of a food, how did it end up in this food? Why is it of concern, and what's method of analysis? All right, so now in the last 10 minutes or less that I have, I will quickly go through the last subject, very short, analysis of extraneous matter. So extraneous matter, anything icky that shouldn't be in your food. These guys. Yeah, so rodent hair, for example, insect wing, a bird feather, more than one wing, I'm telling you, there may be multiple. And then other things that you don't want that are not necessarily coming from biological origin, stones, sand, dirt, metal, glass. So all of these can wind up in your food, and there is no escaping. You know, some people eat insects. So, but insects are everywhere. I mean, when you harvest your wheat, there's no escaping. You're harvesting insects with it. It's everywhere. Also, storage. So think about the storage places. Don't you think there are going to be some rodents there? Sure, there are going to be rodents. And then during transportation. So there might be insects, rodents in the trucks, who knows? But it's everywhere. And during processing, you can, there is no place on earth that doesn't have insects at all, no matter how much you try to eliminate them. So even during processing, we might get some contamination, even though you are doing the good manufacturing, what is it called? Practices, good GMP. All right, so these are the insects that are small and big that will be on your, during storage, you have those little, look at this little mouse that got caught. So there is no escaping. So they put regulations. Everything we eat has some filth. Did you eat your lunch yet? Yeah, there is some filth in everything we eat. That's why we have regulations. The regulations says that you really have to set defect action level. So at which level we say this is defective, we're not going to eat it anymore. So, but we do as much as we can to limit the filth in our food by good manufacturing practices, sanitary conditions, thorough inspection of raw material. You know, when you bring in your flour, wheat, or whatever you bring into your facility, you want to make sure it is within accepted defect action level. So, to give you an example and put a perspective on the filth that we eat, consume. So, look at this. The defect action level for 100 grams of peanut butter is 30 insect fragments and 1 rodent hair. So, anything that is less than 30 insect fragments in, in 100 grams of peanut butter, you can consume it. And you are consuming it. So now look at the flour. It gets even more interesting. 75 insect fragments and 1 rodent hair per 50 grams of flour. So if you have 1 rodent hair in 100 grams of flour, that's okay. Yeah. So they're unavoidable. They're pests. You cannot avoid them. So what do? What we don't eat, we have to eat. Well,
So are the insects inside the kernel included in the insect fragment?
yeah, so this is the milled, when you mill the flour, so the insect is milled now. Well, you know, protein. Yeah. Oh yeah, I'm coming to that. That is a problem, actually. Oh, here's just a joke for you guys. This guy in peanut butter almost notice an increase in hair. So, and this lady here, hold it. This one only has 7% insect parts, but we don't label insect parts, so that's not true. All right, so filth. What is filth? So terminology. Animal, anything animal or other objectionable matter. Contamination. Heavy filth. So heavy filth can be sand, soil, so anything that's sediment. Pellets, excreta pellets. So, you know, they poop too. So what can we do? So we can separate them by sedimentation. There's the light filth. That means, you know, hair, light insect fragments, those wings that we talked about. And then filth, the filth that you can run your, let's say, spices through a sieve, and you can remove some of the little pebbles or pellets or what have you. This is the concern, the question of its number. Yes, it's based on count. So a full insect is one, and a wing of an insect is one, is counted one. So that is a problem when we count. And also there is subjectivity. Some people with no experience, they won't differentiate the insect parts. So some insect parts are very obvious, like this guy's mandible. And then some insect fractions like cuticle from the exoskeleton, you won't know what it is. So, and then there's a lot of transfers, repetitive transfers during the analysis that you might lose some and then count less than actually present. So a lot of the methods is based on counting under the microscope. So spices, run it through a sieve and take what's left on the sieve and count on the microscope per specific gram or 50 grams or what have you. This is an approved method to determine heavy filth in, let's say, nuts. So you use a series of reagents to separate the nut, the shells from anything heavy that would sediment. You filter, you ash, you can either count or you ash at high temperature and weigh, because everything is heavy filth is inorganic. So you can do that ashing and determine. This is a common method, but also involve multiple steps and separations. And this is specifically for a light filth, like insect fragments and rodent hair. You digest, you add different reagents, you add oil, because then the filth, the light filth, the hair, an insect fragment will be in the oil layer. So you have a specific, like a fork that you pull up and you trap the oil up here, and you take that oil and you filter and you put whatever fragments under the microscope and you count. You can do the same thing using separatory funnel. You do acid digestion, you add your reagents and the oil where you have that filth in the oil, and you separate and you count under the microscope. Here are the beautiful larvae, and you do X-ray to see them. And how many of those are allowed per 100 grams? 32. Yeah. So I'm almost done. So you do the X-ray radiology to detect that. You can measure uric acid to determine the presence of excreta. You can do specific ELISA for insect protein. We can do IR to detect later stages of infestation. You can do microscopy, is used for metal types and also differentiating the other glass, plastic, and other. So last slide. So to prevent, we do the X-ray and we count and we get rid of those high infestation. We can get rid of insect fragments in grains by aspiration, blowing air. We can get rid of heavy filth by running our grains on a belt, and the belt has holes and you run air. So the pebbles and heavy filth will stay and your grains will be floating. We can break down, crush the wheat a little bit, aspirate. You can get rid of those larvae, as much of it as possible. You can get magnets to get rid of any metal contamination. All right, fun. That's it. Thank you. Yes. See that question that I went over, the milk, what's in it? There was one question like that. There will be one question similar to that. Okay. Thank you so much. It was a pleasure teaching you guys. I'll see you on Tuesday. Thank you.
Hey Pam, one question.
Oh, yeah, where you going?
Pam,
To me.
hey Pam.
Yeah, Rob.
Um, just I wanted to mention also, I don't know if you got it, but the measuring relative ash to detect diatomaceous earth and or dirt, dust, clay on your wheat. That's something that I had done, and
So, Rob, I can't hear you very well because there's a little bit of commotion in here. So
compare.
if you want to email me because I can't hear anything right now. It's a little bit crowded in here. Oh, let's see. Comparative ash for grass. What's for grazing? What's grazing? I'm not sure. For grain. Yeah, yes, exactly. Yes, yes. So the ash content is very important. Yeah, that's right. I don't have it on my slide, but it's good addition. Thank you.
There's a few more.